And what I'm going to show you is a project that, I mean, we work with phages and phage proteins that target escape pathogens. And for today's talk, I decided to just pull out one of these stories. It's something that's ongoing for the last 2 years or so. And we are focusing on, and I'm going to tell you a story of these three phages that we have that target just one bacterial strain, which is Klebsiella pneumonia. And I know that Klebsiella pneumonia is not a stranger in this room, everybody knows it, but I wanted to just draw your attention to just this particular pathotype, which is the hypervirulent K1ST23 strain. It is different from your classical form, the hospital-acquired infection strain, in the sense that it is community-acquired a strain that is actually very, that would otherwise actually infect healthy individuals. So they would infect anybody, even if they don't have an even if they're not immunocompromised. Clinically, they're associated with liver abscess, meningitis, and they actually cause an infection in the inner eye. So that's quite difficult to treat. The first reports were from Asia, in Southeast Asia, but we know that it's been spreading globally. The strain that we are currently using was isolated in Germany, in Leipzig, so we know that it's been going around. And in terms of virulence factors, the one that I would really like to draw your attention to is this mucoid phenotype. So it produces a lot of capsule. It's got a very strong capsule production. And in terms of the string test, is what you see here. It's a positive string test. So it's very slimy, actually. Historically, this strain is actually, in a sense, easy to treat because they are usually susceptible to antibiotics. But we've also started to see an increasing number of reports where these strains have actually started acquiring resistance as well. So this has been quite alarming. So making use of the infamous double agar overlay method, we kind of isolated three phages. We got them from sewage water that was taken from all around Leipzig, so just around the city. You can see that plaque morphology, they're all quite distinct. So in terms of isolation and purification, it helped to make it a lot easier. Two of them are C4-like viruses. So we called them spear and loop. And because the last one was a proto-like virus, we called it shorty. And from these, we then went on to genomic characterization, which is what you normally do, right? So we isolated the genome, sequenced them, built the genome, and we see that they're all very similar in size. So they're about 40,000 base pairs. They're strictly lytic. They have close relatives to other phages around that's been isolated around the world. But strikingly, even though we isolated them from pooled sewage water around the same city, we realized that they're not actually, they're actually quite distinct. So when you try to align their genomes, they don't align at all. You find no homology. If you were to try to put them in the same tree, they don't cluster together either. So they are really distinct phages. So we decided, instead of looking at what sets them apart, maybe the better way would be to see what they have in common. And what they have in common is the host, right? So we started pulling out tail fibers and tail spike proteins. And we analyzed these sequences in two ways. One was to look at just alignment, sequence homology, which brought us to a dead end because there was no homology. So then we looked at structural homology. Now we know that for most phages, tail fibers and tail spike proteins would form a trimeric complex. And we did that in silico, so we made use of AlphaFold and Chimera X to build these structures. And we were actually quite happy to see them because if you were to look at, for example, spear, the tail forms kind of a structure that looks very similar to the tail of the phage. So that was something promising. We were quite happy with that. What we also saw was that two of the phages, for loop and shorty, the tail spike proteins actually align with each other structurally. If you were to superimpose them together, you'd find that both of them actually superimpose and they form, they actually do have the same structural homology. And when we aligned this to just the protein database, we found that it also aligns to the K1 specific lyase domain, which was very nice. So all this data was coming together. We wanted to further validate that perhaps the binding site of, well, perhaps the binding target would be CPS or the capsule. So there are several ways to do this. We chose hopefully a more non-biased way of doing this by generating phage resistance. So we generated phage resistance against loop. We just picked one of the phages. We isolated 25 colonies, and making use of the double agar overlay method, we then tested to validate that they're resistant. So the top panel is for wild type, and you can see that both phages would infect. If you use one of the resistant mutants, you have resistance for loop, but you also see the same resistance for shorty, which was quite nice, which is what we expected. What was really interesting here was that every single one of these phages, all 25 colonies, lost their mucoid phenotype. So from being mucoid, the mutants were suddenly non-mucoid. And we thought this was a really nice, interesting result. We also sequenced these mutants, and we did see that the mutations that were generated affected every single gene along the capsule synthesis pipeline. So that was really quite nice. To further validate this, we then also looked at attachment assays. In this sense, we made use of equalize a negative control, and we saw that, okay, we've got none of the phages would bind E. coli, which is nice. If you were to look at one of these resistant colonies, one of these resistant mutants, you also don't see any binding. And with the wild type, you do see this binding. And in this assay, we decided to include SPEAR as well. And this was nice to see because it looks like SPEAR is also using CPS or the capsule as one of the receptors. So we went on to also look at the resistance profile for this. That is just the wild type as well. And you see that, okay, SPEAR and loop, they infect. If you use the non-mucoid variant, you lose binding of loop, but you also lose the infection for SPEAR. Now we decided to shake things up a bit, so we decided to isolate phage resistance against SPEAR. And this was where it got a little bit tricky because we saw that phage resistance against SPEAR, well, you see the resistance, that's great. But when we tested loop against it, you still can see infection. So it seems like loop does use the capsule for binding or for infection, but it also uses something else. So you can see that both phages use the capsule, but one of them actually uses something else on top of this. It could use something else. So we thought, okay, well, if we want to, let's say theoretically, hypothetically, because you can't use phages for treatment in Germany, if you want to use them for phage therapy as a cocktail, and you know that both phages don't really use exactly the same receptors, if you put them in a cocktail, what happens? Do you get any advantage? And so that's what we did. We put them together, and we saw that both phages were able to suppress bacterial growth for about a period of 7 hours. And then we start seeing resistance coming up. Now, this is exactly what we see when we use both phages individually. So if we were to just look at loop, or we just look at SPEAR, they also suppress bacterial growth for a period of 7 hours, and then you see resistance coming up. So it's exactly the same. So there is no actually advantage in this sense. And we thought, well, okay, but what's happening in this cocktail? Are they acting synergistically? Are they acting independently? Or maybe there's antagonism. So to look at this, we looked at the bliss independence assay, which is basically a statistical model. If both phages work independently, then the values would be a 0. If they're positive, then they're working synergistically. But what we're seeing here is all these values are actually in the negative indicating that they're actually antagonistic, which kind of makes sense if you think about it, because they're probably competing for the same receptor. And you can see that if you add more phage, then it becomes even more, you see even more antagonism. So it was quite a nice, well-rounded thing. So from this, I decided I think I'll just stop here before it gets more complicated. What I've shown is that we've got three lytic phages. They are genomically distinct. All three phages target the capsule, but we also know that loop probably uses one more. Phage resistance seems individually on a cocktail, phage resistance appears after 7 hours in vitro. And if you use them as a cocktail, it seems that Phage and loop actually work against each other. And in such cases, maybe just using them as single phages would probably have the same effect. So probably it's best to know your phage, to know your receptors before using your phage. So with this, I'd like to thank the people in my group. All the work is done by just one PhD student, so this is Johnny Bex Alpiv. And I'd like to thank you for your attention. I'll be happy to take questions.